This conference is a lot about cell and gene therapy, but there is also about some other things. So I will try to entertain you for a little while about something that I think is very exciting, and that is to personalize organs and how we can cure diseases by doing that. So my name is Petter Bjorquist. I'm the CEO of Verigraft in Gothenburg, Sweden. You see a photo of our headquarters in central Gothenburg. We are a classical university spin-off. We are privately funded by investors, and we have a highly experienced management team that has been around for a long time in this space. We are today talking about advanced therapies and how we now can help and contribute to expanding this very interesting field. So to start off, I will give you a little personal reflection of what I'm talking about today. You see something that we have in the waters around Sweden. It's a lobster. You can also see that this lobster has one big and one small claw. And what I'm trying to illustrate is that lobsters are very good in regenerating themselves. So if you break off a claw, there will be another new claw growing out. Unfortunately, if we are unlucky and losing an arm, there will not be another arm growing out. So when it comes to us humans, we need some help in regeneration. So therefore, you see more and more often these type of pictures, but be careful. This is not so rocket science as you can imagine. You can create with a printer, for example, something that looks like an ear, but promise, don't think this will help you hear better. But a new heart is a good thing, right? And you see also these type of things. And don't misunderstand me. I'm a strong believer in bioprinting and 3D printing, but it's still a couple of years ahead until we can see a heart being printed and being implanted. What can we do in the meantime? I think we can do something without creating new organs, but we can change and modify organs and making them clinically useful. And that is what my presentation is about. So what type of problem am I trying to solving then? It's all about transplantation, and as you know, there is a lot of transplantation ongoing. So things like an heart is of course transplanted today because we will die if we don't have a new heart. But other organs like a blood vessel is not very often transplanted for a very simple reason. To make a transplantation, you need to get rid of the immune system, or at least press it down to almost zero. And that is something you have to do for the rest of the patient's life. So by saying that, you may understand what I'm out for. If we can transplant things without suppressing the immune system, we would have a great medical benefit. So can we do that today? Yes, and I will show you how. This is illustrated by a blood vessel, in this case a vein. So we take a vein from a donor. We take off everything that tend to cause an immune reaction, being cells and DNA, in a process called decellularization. What is left then of an organ? It's a lot left. It's a scaffold left, and almost all organs look pretty much the same if you take off all the cells, because the cells are a lot about function, but the three-dimensional structure is other things beyond the cells or behind the cells. It's a network of proteins, and we take advantage of that. So this scaffold can easily be frozen for years in our freezers. And then we can apply our second process, the personalization process. And this is something that has been around for many, many years. And typically, others are using pure population of cells for personalization. And that is in one way excellent, but in one way very time consuming, very expensive, and regulatory wise it's a lot of questions around culturing cells for a long time in vitro. So instead of that, we are not using any pure population of cells. We are using blood from the patient. So we take a blood sample, only 50 ml. We mix this blood into a personalization solution with some tricks that we have in my company. And we apply this for a process that makes a new identity to the scaffold or to the organ or the tissue. And this personalization is only 1 week long. Instead of many months, a lot of costs, and a regulatory questionable process, we do this very lean and efficient. And out comes something that can be transplanted and will be seen from the patient as something autologous-like instead of foreign. So does this work? To cut a long and many years story short, this is a few slides from our most advanced large animals. So these are pigs that we have been running for 1 year. And as you can see from the left, perfect blood flow after 1 year. We open up the vessel that we have transplanted, the personalized vessel. You know, you can also personalize a pig vessel by taking material from different pigs. And we have this surface lined by very nice endothelial cells. So what are we aiming for? We are, of course, having a first indication in sight. And our first indication is this one. In our groin region, we have blood vessels or veins that are looking like this. They are working because we need to pump up the oxygen free blood from the feet up to the heart. And as you can see, there are a couple of valves or a pair of valves that are opening when the muscle pump is pumping up the blood, but closing when the blood are trying to go down by gravity to the feet again. If we don't have these valves are constructed by evolution for some reason. If they are failing, you are in a state of chronic venous insufficiency. And for the patient, it's starting with pain and ulcer. But unfortunately, this disease is ending up with very severe bleeding leg ulcers for most of the patients. So this is severe CVI, not very nice, as you can see. And we want to do something about this because there is nothing done for these patients today. We are, of course, taking care of the wounds, but we can't treat this disease, or rather, we can't cure the disease. So this is our product. And as you can see, it's a pair of valves that are working, and it's personalized. So in our first generation of products, we take this material from deceased donors. So a deceased donor is giving us this, we are personalizing this, and we are transplanting it. So let's see. This is just a little thing to show you that such a piece of a vein is working. You can clearly see the valves, if you look carefully, that are opening and closing while the heart is pumping and the muscle pump is pumping. So we are now at clinical stage with this. So we have a first phase 1/2 clinical trial, a first-in-man trial ongoing in Spain. That's very nice to say here in Barcelona. So in Spain, we have now the aim of recruiting 15 patients in this trial with severe chronic venous insufficiency. So this is the first of its kind worldwide, this trial, and we hope this will be a game changer. So first patient, this is very new data. So the first patient we included just a few months back, and we have recently run the safety follow-up for this first patient. And luckily enough, we see improvement and we don't see any safety concerns. So we are continue this trial. And the aim is, of course, to go to the market in Europe. In discussions with the regulatory experts, we expect some 70 to 80 patients to go to the market, first market approval in Europe. And we are using our collaboration network for that. In the US, we are, of course, also very eager, but we have decided to first see safety and efficacy in Europe before we make a full show in the US. But that will start towards the end of this year, we hope. So support for a business model. We are a company, as you understand. The reimbursement model, I can tell you that a typical patient with CVI is on sick leave, even early retirement, a lot of cost for taking care of the wounds. So it's a very high burden for the society. So our health economy calculations are very positive. And we, of course, calculate our cost of goods. And remember, our process is very lean and quick and cost efficient compared to other peers in this area. Regulatory-wise, we are following this and see how we are taking the route to the market. It's regulated as a classical ATMP, as most of you know. Just to mention and tease you, we have in large animals 2 other projects. We have arteries, which is it's an even bigger area for commercialization of a technology like this. because there are some very demanding indications around in this area. We also personalize the longer peripheral nerves in your extremities in the same way, and we have also run successful large animals with this product. So, of course, these are big markets, and just to tease you once more, and this is maybe a bit interesting, I hope you think, because today we are using, as I described, human-donated material, and you can think that maybe we are running into limitations in the future. Do we have that many human donors that are willing to donate? We have quite a few, we think, at least in the westernized countries. But we think also that we have other donors, and animals can also be donors, as you may know. So xenogenic tissue, being a blood vessel from a cow or a pig or a sheep, can in the future be used as a scaffold for our products. We have also these type of studies in large animal studies. But for the future, there is something that I'm sure you are also excited about, and that is printing. So we have projects ongoing where we are going to print a scaffold. Then we are totally independent of donors. So that is a very good future. And this is just a recent project slide where we have received money from the Swedish government to work with a few very good partners. In the middle, we see the Research Institute of Sweden, and we have Cellink as an expert in 3D printing, bioprinting, that will help us with the printing part of that project. We are, of course, scaling up. You don't have to look too careful, but you may also think that creating organs or tissues, how can that be scaled up? And we have several projects going on where we are scaling up to a decent level with this. Sweden tried to be one of the most active countries here. We have several governmentally backed bigger projects where we think that Sweden can take a leadership role in the future. So CAMP stands for Center for Advanced Medicinal Products, and it's a Swedish governmentally backed big project. My company is also backed by some important granting bodies. In Europe, we have the EU programs, and the most important is the previous SME instrument granting possibilities. You can see it's quite a bit of money. And we have other, like Eurostars, but we have also many Swedish governmental grants around. So there is more than me personally that believe in this. So back to my lobster, I want to wrap up and thank you for your attention and say that together we have the possibility to make some new good things and regenerate part of the body that we didn't know before that we could do. And even if we are not lobster, I hope that we could regenerate more organs than blood vessels in the future. And my final words is that this is our logo, and we have just started fundraising, so should it happen to be someone around with contacts in that area, we are happy to discuss how you could join our exciting journey. So thank you very much for listening. It's a pleasure to be here.